All right. Sam, whenever you're ready.
Thanks, Anne. Good morning, good afternoon, good evening, everyone, depending on where you're joining us from. And thank you for joining us. Welcome, everyone, to our third annual Shine a Light on TANGO2 celebration. For those of you that I haven't had the pleasure of meeting yet, my name is Sam Gregoire, and I am a volunteer and the chair of the TANGO2 Research Foundation's fundraising committee. More importantly, I'm the proud Aunt Sammy of 5-year-old Jake Taggart, one of our TANGO2 kiddos. Today, we are celebrating the accomplishments of our third annual virtual fundraising campaign. As we continue through our presentation, feel free to use the chat box and the reactions on Zoom to help us celebrate today. Shine a Light on TANGO2 is a campaign that was inspired by Kasha and Mike Morris's banquet in Portland, Connecticut back in 2018. That inaugural event raised $33,000 with 175 attendees supporting their son, Ryan, and our research foundation. Fast forward a few years later, we'd grown into an international community and the COVID-19 pandemic hit. So just like this resilient community, we pivoted and blossomed into a virtual campaign with over 700 donors. Today, we celebrate our community and campaign that kicked off in February during Rare Disease Month. Our teams have worked very hard to raise money for life-changing research, and we are here today to celebrate all of your accomplishments. By coming together to support our mission, we are shining a light on TANGO2. Every dollar raised holds promise to a bright future for our children and young adults living with TANGO2. During this year's campaign, 22 families were represented all over the world. They created fundraising teams and pages that represented our heroes, those living with TANGO2 deficiency disorder. We were joined by more families than ever before and from different countries. Together, we raised an incredible amount of money for our mission to fight TANGO2 deficiency disorder. We set a $100,000 goal and proved pretty quickly that we are going to hit our goal and help fund life-saving research to support our children. Now, I would like to introduce you to the treasurer of the TANGO2 Research Foundation's board, Kara Cameron, who will take a moment to celebrate our fundraisers.
Hi, everyone, and thank you for joining us today. And thank you, Sam, very much. So this year was our best year yet, and I am so happy to be able to celebrate all of our participants. Bria's bright light and story of survival continues to inspire everyone around her. And because of that, we doubled what we raised last year. Our family and friends had a Mother's Day fundraiser, and Bria's dad, Matthew, had a tremendous outpouring of support for his raffle events. Together, our teams raised over 30,000 Canadian dollars. Team Ryan also had some heavy hitting fundraisers, with Kasha selling beautiful flower arrangements from her yard and Mike generously donating a portion of proceeds of his business to the foundation. Lynn held her restaurant fundraiser at Popular Pose Restaurant once again. Our funders from Down Under proudly represented the children of Australia. David was all smiles as he successfully ran a half marathon and grew his team to 5 members this year. Congratulations, David. Veronica held her raffle for Thea again this year. She had set a goal to raise $4,000, but ended up raising almost $10,000. It's amazing. And she continues to be top fundraiser year after year. Our UK families were shining bright this year as well. Sebi had his very own art exhibit, and his mom, Amanda, climbed Mount Kilimanjaro, 3 peaks over the course of 3 days. When Otis turned 3 this year, his mom, Sheehan, set a goal to do 100 squats a day for 28 days during Rare Disease Month. That is a total of 2,800 squats. Not something I could ever do, so congratulations. Phoebe and her friends and family really knocked it out of the park for their first time fundraising. Their family friend, Pete Blunt, raised $7,000 US after running a marathon in an amazing 5 hours and 25 minutes. Absolutely incredible. However, Phoebe and her supporters are not done yet. They have a roller skating fundraiser planned for July 8th and more to come. Team Italy did a great job this year. Anna raised money for Lorenzo, and Giorgio and Rashmi still found several new supporters for their son Eduardo. Their friend Gloria, who was celebrating her 60th birthday, asked for donations. There was even a rock cover band raising money. The New York crew came back for a third year in a row and was as creative as ever. Tyler's crew had their annual golf tournament, and Tyler's nephew made fresh kunkel cookies. Very cute. Team Katie Clay teamed up with Dr. Mackenzie and had restaurant fundraisers and a big yard sale where no one could resist the adorable kids raising money for a good cause. Speaking of yard sales, the Mitchell family held one too and rounded up both grandmas and aunties to help out. Robin and her family raised money and also donated to several fundraising pages. Our families in Texas continue to show up strong. The Moore family for Ellie and Team Sammy's Bravehearts continue to impress us. Marcy brought back her most popular fundraising events, Raising Canes and those tasty tamales. The Wiley family continues to fundraise and fight for Aislinn. This year, we were so excited to welcome Team Brazil, led by Diane. Diane, who represented all of these beautiful faces. Thank you so much for joining us for the first time this year. And of course, all of those who supported our ambassador family, the Friars. Kelly held a tango ballroom dance event for her sweet nephews, Hudson and Cole, and a tea event where she had a whole community support her. Caitlin and Russell held Hudson's Heroes 5K annual run once again, where there were even runners participating virtually, and a bar workout fundraiser at her local studio. Team Fryer Brothers, Cole's cousins, and Christina Barber also showed up strong for Hudson and Cole. Caitlin and Russell, from your moving video about your family to the nonstop fundraising you did, thank you for sharing your story, inspiring us all with your commitment, dedication, and resilience. Thank you so much to everyone, and congratulations to all of the teams. And now I would like to pass it over to our amazing 2023 Shine a Light on TANGO2 campaign chair, Kelly Hernandez.
Thank you, Kara, and thank you everyone for joining us today, and for everyone who worked so hard to fundraise for this year's Shine a Light on TANGO2 campaign. Now I'm going to take a moment and recognize our top teams and participants. Congratulations to our top 5 individual participants: Kelly Hernandez, Mike Morris, Veronica Jones, Matt Dort. And our top individual fundraiser for this year is Kara Cameron. Kara raised $15,785 Canadian dollars. Amazing. Congratulations, Kara. And now we would like to recognize our top 5 teams. Congratulations to Cole's Cousins, the Dorts, Team Bria, and Fryer Brothers. And the #1 team is Team Ryan. Team Ryan raised $16,709 US. Thank you, Mike, Kasha, and Lynn for stepping up and inspiring all of us to continue to share our stories and be a voice for our children. And now I am so pleased to announce the total amount we've raised during this year's campaign. Together, we surpassed our $100,000 goal and raised $108,830. Yay! I'm so proud that our small and determined community accomplished this, especially as we were able to surpass last year's results. With this money raised, we can continue to fund research that will lead to treatments and a cure for TANGO2 deficiency disorder. I also want to take a moment to thank our ambassador family, the Fryer family. Thank you, Caitlin and Russell. For sharing your emotional and heartfelt story with us and giving us your time and energy. I know it was not easy to share Hudson and Cole's story, but I hope that seeing all of these people come together to support your family and other TANGO2 families will allow you to feel a sense of hope. I know I have never been more moved and inspired. And now I would like to introduce Pariya Sadi. She is a student currently pursuing an undergraduate degree at Concordia University in Montreal, Canada. Recently, she won 2 awards for her work on vitamin B5. First, she was awarded the Farag Foundation Award for Best Undergraduate Honors Project. She also won the Best Poster Award in the Concordia Health Sciences Symposium, where her category included undergraduates, master's, and PhD students. Pariya would like to say a few words.
Thank you so much, Kelly. Hello, everyone. My name is Pariya, and approximately 2 years ago, I had the opportunity to join Dr. Satcher's laboratory, where I embarked on a journey to apply my knowledge within a research setting. It was during this time that I was introduced to TANGO2 project, which was an exploration into a protein with limited available information. Over the years, the number of diagnosed cases of TANGO2 mutation has been gradually increasing, even though it still remains a rare condition. From the very beginning, this project captured my interest, and I found myself captivated by the scientific interests it presented. After devoting a year to this project and coming up with a potential therapeutic approach, I had the privilege of attending the TANGO2 Family Conference held in Orlando. It was during this event that my life took a profound turn. I realized that TANGO2 represents much more than just a protein. It symbolizes the immense potential of science to positively impact the lives of those greatly affected by it. Witnessing the resilience in the eyes of TANGO2 warriors and engaging in conversation with their families enlightened me about the profound rewards inherent in this research, rewards that simply cannot be quantified. What I would like to say is that you're not alone in this fight. Although the path may feel uncertain at the times, advances in research and medical settings is being made. Scientists in my lab and other around the world and medical professionals are tirelessly working to deepen our knowledge of TANGO2 deficiency disorder, leading to improved treatments and potential backgrounds. While no one can predict the exact timeline, progress is being made. And they remain steadfast in their commitment to finding solutions. These past 2 years were the most rewarding and challenging of my academic journey, and I have enjoyed every moment of researching and working on TANGO2 project. However, that has truly fueled my passion and dedication throughout this endeavor is keeping the families affected by TANGO2 at the forefront of my mind. They are driving the force behind our research, and their stories and experiences have deeply touched me. I would like to say thank you, Dr. Michael Satcher, Dr. Gambari, the TANGO2 Research Foundation, and all the families for this incredible experience. I'm forever grateful for the knowledge gained, the connections made, and the inspiration you've provided. Together, we will continue to strive towards a beautiful future for everyone touched by TANGO2.
Wow, thank you so much, Pariya. This means so much to us, and your words are so heartfelt and sincere, and we're truly just so lucky to have you be a part of the TANGO2 family. For those I haven't had a chance to meet yet, my name is Ann Geffen, and I'm the Executive Director of the TANGO2 Research Foundation. I want to first thank you again to all of our fundraisers, and of course to Kelly Hernandez for really being a fantastic leader for us this year. If you would like to continue supporting us, you can donate to one of our teams still by going to www.shinealightontango2.com. And this site will be open until the end of the year. We also have a few very important announcements. First, please know that we're continuously looking for volunteers to join us. We hope that you had a great experience at this campaign this year. Please feel free to email me at ann.geffen@tango2research.org. And I would love to hear how you would like to get involved. We can't do the work without volunteers, so I hope to hear from you. Please also mark your calendar for our next TANGO2 talk on September 16th at 10 a.m. Eastern Time. We have a dietitian, Brandy Rawls-Castillo, who will talk about nutrition for individuals living with TANGO2 disease. We also have our upcoming virtual TANGO2 Research Symposium on Friday, October 27th at 9 a.m. Eastern Time. We will have more information and details coming, but for now, please make sure to mark your calendar for that. And finally, please save the date for our in-person TANGO2 Family Conference next year from Sunday, June 23rd through Tuesday, the 25th, next year, 2024. I'm so pleased to share that we're going back to Orlando, Florida at Disney's Coronado Springs Resort. And now we are very excited to announce our Cycle 3 research grantees. But first, I would like to introduce you to our wonderful research engagement director, Dina Chisholm. Dina has been with the TANGO2 Research Foundation for almost a year now and has been instrumental in helping the foundation strategize to meet its research goals, support our vision, and fulfill our mission.
Thanks, Anne. Hi, everyone. I'm so thrilled to be celebrating a successful Shine a Light campaign with each of you. It was such an honor to be a part of such an amazing event. Job well done to each of you. I'm so very pleased to report that after combing through several letters of intent this year, we've invited 7 applicants to submit full research grant proposal for this TANGO2 Research Foundation Cycle 3 research grant funding. After a very rigorous grant review process with formal reviews from our TDD academia and clinical professionals, as well as our TANGO2 Research Foundation Research Committee, we are excited to announce we'll be funding 3 of the 7 highly competitive proposed projects. With your support, we were able to fund research totaling $110,000 this cycle. That's amazing. Each researcher will begin their projects July 1st, and we're really extremely optimistic about what their project findings could mean for our community a year from now. At this time, I'd like to turn it over to Bev Ptolemy. She'll introduce our first TANGO2 Research Foundation Grant Cycle 3 awardee. Bev?
Hey, I'm Bev. Nanny to Daniel Smith, who is son to Talia Ptolemy, who is part of all of this. It's my pleasure to introduce our first TANGO2 Research Foundation Cycle 3 Research Grant awardee, Dr. Vandana Gupta. Dr. Gupta is being awarded in the amount of $25,000. Dr. Gupta is Assistant Professor of Medicine at Brigham and Women's Hospital in Boston and Principal Investigator of Gupta Lab. Dr. Gupta's laboratory studies the genetics of neuromuscular diseases. Her research group has identified several new genes in neuromuscular diseases and developed animal models to test novel therapeutics and study disease pathophysiology. Sorry, it's a bit late here in Australia. Her group is currently developing therapies for rare neuromuscular diseases for small molecule screening in cellular and animal models and gene targeting approaches to develop cures for patients affected with those disorders. Her grant funded research entitled Metabolic Genetic Modifiers of TANGO2 Deficiency Disorder will focus on identifying the genetic, molecular, and phenotypic changes in varied clinical presentations of TANGO2 deficiency disorder in zebrafish models. The aim of this research is to improve TANGO2 deficiency disorder management and therapeutic options for our TANGO2 affected children. And a very big congratulations on your award, Dr. Gupta. Thank you. Good? Perfect. Okay,
so I'm, oh, can you hear me okay? Hello?
Yes, I can hear you, Hannah. Yes.
Oh, sorry. Hi, I'm Hannah Driffel, parent to Phoebe. And we've been on our TANGO2 journey with Phoebe since March, when she was diagnosed at the age of 6. And I'd like to say I'm so grateful to this community and very pleased to introduce our second TANGO2 Research Foundation grant awardee. So Drs. Lele Zahang and Alexander Baijie, who are being awarded $35,000 as an award. Dr. Zhang is an assistant professor of molecular and human genetics, molecular physiology and biophysics, and internal medicine at the Baylor College of Medicine. She is the chief of the cardiovascular genetics clinic at Baylor College of Medicine and is board certified in clinical genetics and in internal medicine. Dr. Zhang is a physician scientist, and her laboratory studies the impact of gene regulation on the health and disease of the heart. More recently, the lab focuses on using induced pluripotent stem cell differentiated cardiomyocytes combined with genome editing tools to understand inherited cardiac diseases and translate these findings to the bedside. Dr. Baijie earned his PhD at the University of Belgrade in 2011 and became a postdoc at Baylor College of Medicine in 2012. In the past 11 years, he's exclusively worked with the human pluripotent stem cells as cellular disease models, being devoted to the pursuit of innovative neuroscience research with significant translational value. In 2018, Dr. Baijie was appointed the Director of Human Neuronal Differentiation Core, which was designed and built during 2017 under his guidance. His core is engaged in expanding the set of methods used to examine human disease phenotypes in vitro, including establishing novel molecular and biochemical assays, implementing genome editing for correction of potential disease variants in cellular models, and advancing single cell and network level electrophysiological and live cell imaging. Dr. Zhang, would you like to say a few words about your research?
Hello.
Good morning, Dr. Zhang.
Hi, congratulations everyone for doing a wonderful job in fundraising. I'd like to thank the foundation as well as all the families to give us the opportunity to do work in TANGO2. We started involved in TANGO2 disorder back in 2019, studying the pathogenesis of the arrhythmia occur in TANGO2. We use patient heart cells to study why people with TANGO2 disorder have arrhythmia, and we made great progress and have found that folate can potentially terminate this lethal arrhythmia. I attended the TANGO2 family meeting in Orlando last year, and from talking directly to all the families, I learned that the most concern for everybody is the neurodevelopmental disorder. So I'd like to do something in that aspect. So together with Dr. Bajic, we decided to take advantage of all the skill sets and resources and platform we already generated to study patient-derived brain cells, the neurons, and different forms of neurons, and also use mouse models of TANGO2 to study the neurodevelopmental behavior. In particular, we're interested, as most of the TANGO2 family are interested in finding out what is the role of vitamin B in all the presentations of TANGO2 disorder, including the neurodevelopmental disorder, the seizures, all the problems we're dealing with every day. So that's our goal, to use the best models we can imagine, the human brain cells, as well as the mouse in vivo models, to study the effects of vitamin B in TANGO2 disorders. I thank all of you to give us the opportunity. You are the true inspiration to direct our work. I hope someday we can make an impact in understanding and treating TANGO2 disorder. Thank you.
Thank you so much, Dr. Zhang, and to you both, and congratulations on your award again. And I'd now like to turn it over to Giorgio.
Thank you, Hannah. Hello, everyone. My name is Giorgio Pochettino. I'm the parent of Eduardo, a 9-year-old boy with TANGO2 disease. I will be introducing our last and final TANGO2 Research Foundation Research Grant, Cycle 3 awards. In particular, Dr. Michael Sacher and Chiara Gambari, who are being awards 50,000 award. Dr. Michael Sacher is a professor in the biology department at Concordia University and an adjunct professor at McGill University in the Department of Anatomy and Cell Biology. His laboratory focuses on mechanisms of membrane transport between cellular compartments and diseases related to defects in this process. Work which began as a research associate at Yale University. He discovered the trap complexes in the yeast and identified mammalian-specific trap proteins. Using genetic, biochemical, structural, and cell biological approaches, his group showed that they play pivotal roles in membrane transport and have linked mutation in a number of trap genes to human disease. His laboratory recently began characterizing the function of TANGO2 using a variety of model systems, demonstrating a role in both membrane transport and at the mitochondria. Dr. Chiara Gamberi, a molecular biologist by training, became interested in how molecular interaction affect the whole organism and embraced development biology. As independent researcher in Canada first, and now new faculty at Coastal Carolina University, she and her group study how gene expression is regulated in space and time in living organisms in health and disease. They employ the genetically tractable fruit fly. The name is Drosophila melanogaster. They combine genetics, molecular, and biomechanical assay, omics approaches, and innovative whole animal pharmacological assay for chemical probing and fly-powered drug discovery. They are applying their know-how for modeling polycystic kidney disease, PKD, the name, and kidney cancer, and they fly towards unraveling the mechanism of TANGO2 deficiency disorder and designing effective therapeutics. Sensitive to the inclusivity, Dr. Gamberi has developed new research pedagogy to teach undergrads how to research scientific information, elaborate and write manuscripts for peer-reviewed publication. The Sacher and collaborating Gamberi laboratories have been studying the function of TANGO2 in 3 different model systems, including human cell, fruit flies, and yeast cell. This research, a lipodomic analysis of multiple model systems to better understand the role of TANGO2 in lipid homeostasis, will focus on identify which lipid species are alterated and which lipid pathway are dysregulated in absence of function TANGO2. Employ combined molecular and genetic analysis in 3 model systems. Although several lipid species have been found to alter the human cell devoid of TANGO2. A comparative analysis in 3 model systems will facilitate identification of the key lipids and pathway involved in TANGO2 disease. Several hypotheses as to how TANGO2 regulates lipid homeostasis will be tested from biochemical in vitro with purified components. Dr. Gamberi and Sacher are confident that the proposed integrative research will make important strides in closing in one of the roles of TANGO2 in lipid homeostasis, a function likely to be dysregulated in disease state. The study will also shed light on how vitamin B5 affect the lipid profile and may suggest alternative or additional treatments option. Dr. Sacher kindly recorded a message for us today. Let's see the video.
Hi, everyone. Sorry I'm unable to be there in person today, but I do appreciate having a few minutes to talk about the project that was funded by the TANGO2 Foundation in this in this cycle 3. So let me just share my screen. I'll present a few slides here to give you an introduction about what it is that we'll be doing. So before I start, though, I would like to first thank the TANGO2 Foundation for funding this Cycle 3 project and the confidence that they have in our abilities to move TANGO2 research forward. And that would not be possible if it weren't for the donations that come in very generously to this foundation. And so I think you've heard about specific people who have coordinated that in various countries throughout the world. And so those efforts are needed and appreciated in order to allow these types of projects to be funded. So this is the title of our project that's funded. I'm going to come back to that title at the end, just a couple of minutes here. So in a couple of minutes, I'll come back to that title, and hopefully it'll make a lot more sense to you. It's pretty scientific right now. There we go. So we know that mutations in the TANGO2 gene lead to what we're now calling TANGO2 deficiency disorder or TANGO2 deficiency disease, TDD. And in work that was really spearheaded by Priya Asadi, and you've heard from Priya already, and I think she probably was overly humble and didn't really toot her horn nearly as much as she should have. But Priya did fantastic work to show in a fruit fly model system that supplementation of TANGO2 deficient fruit flies with vitamin B5 had profoundly positive effects on the behavior of those flies. We also carry that work out in fibroblasts, skin cells, from an individual who is TANGO2 deficient. And so we know it's not just fly specific, that vitamin B5 is having this positive effect on TANGO2 deficiency disease. What we don't know is why this happens. So everything between TANGO2 deficiency disease and why vitamin B5 is actually helping is a big black box. And we hope in this project to start opening that black box a little bit and seeing a little bit what's inside. So the work that we're going to be doing in this project is going to, and work that we've already done leading up to this, has been mainly done in 2 model systems: the fruit fly, again, spearheaded by Priya, and fibroblast cells, skin cells that were taken from individuals. And what exactly does vitamin B5 do in a cell? Well, there's a very highly conserved 5-step pathway that converts vitamin B5 into a small molecule called coenzyme A, or CoA. That's terrific. What does CoA do? Well, CoA can bind to fatty acids. And so it can be thought of as an activator of fatty acids. And what does this CoA fatty acid entity do? Well, it does many things in a cell. It's largely used to generate lipids that are in cells. These lipids are found in the membranes of many organelles in the cell, of all organelles in the cell. Okay, so if vitamin B5 is positively affecting TANGO2 deficiency, that would suggest that TANGO2 has something to do with lipid metabolism. And that's our main hypothesis, that TANGO2 is involved in lipid metabolism, and the absence or deficiency of TANGO2 is going to result in an imbalance of lipids that are found in the cell. Now, this rather complex graph here is just to illustrate that we've already started to look at this, and our hypothesis is holding up. And what we're looking at here is, in a very complicated way, is showing you lipids that are either overrepresented in TANGO2 deficient cells. Those are shown in red, and you can see there's a lot of them. And in blue dots are lipids that are underrepresented in TANGO2 deficient cells. Normally, if everything was the same, we would expect to see only black dots between these 2 vertical lines. We don't. We see a lot of red, a little bit of blue, suggesting that there is indeed a lot of lipid imbalance that's going on in the absence of TANGO2. So how are we going to tackle this? Well, we know that TANGO2 or TANGO2-like proteins are found in many different organisms, not just in eukaryotes, but in lower organisms as well. That would suggest that the function of TANGO2 is highly conserved. So now, and this is what we're going to be doing in AIM-1, is if we just take a sampling, look at the lipid profile in a sample of different organisms, including flies, including human fibroblasts, including yeast cells, and perhaps some other organisms as well. What we hope to see is some sort of a common denominator, meaning a lipid or a class of lipids or several lipids that are overrepresented or underrepresented in the absence of TANGO2 across all of these various organisms. And those will be the ones that we want to focus on. All these other lipids may be imbalanced because of some secondary effect, but the primary effect of TANGO2 may just be on a subset of lipids. And by looking at various organisms, we hope to find what those lipids are. And in the second aim, which is where the expertise of Dr. Gambari comes in, is to use fruit fly genetics in what's called a genetic modifier screen. There's a particular phenotype in the flies that have a deficiency in TANGO2. And if we combine that with a mutation in another gene, we may exacerbate that phenotype or alleviate that phenotype. And that's going to start telling us in a genetic way what pathways may be affected in the absence of functional TANGO2. So that's in a nutshell what the project is. And if we go back to the title again, so a lipidomic analysis, now you know, is just looking at the lipid profile of multiple model systems because of the conservation of TANGO2 across various species. And that will help give us a better understanding of the cellular basis for the vitamin B5 therapy, which is alleviating the defects or is alleviating the phenotypes seen in the TANGO2 flies and in TANGO2 deficient human cells. And so we hope that we're going to start opening up that black box, having a little peek inside, and getting much better, or getting a slightly better understanding, at least in the short term, and then in the longer term, hopefully others can contribute so we can just throw the cover off of that black box altogether and know for sure what TANGO2 is doing. So once again, I thank everyone for their contributions to the foundation, and I thank the foundation for funding the project that Dr. Gambari and I have submitted. And I hope you enjoy the rest of this TANGO2 event. And I hope to see you in future TANGO2 events in person.
Thank you very much, Dr. Sacher. I would like to ask Dr. Gambari if you would like to say a few words. And thanks
Hi, everyone.
for taking part.
Sorry.
Yes, thank you. I think you gave a very thorough introduction, so there is very little to add scientifically speaking, together with what you said and Dr. Sacher. But one consideration that I would like to do today is that I particularly appreciate how this event is put together and how this foundation is working. So bridging and keeping open communication between the patients and their families who are, affected by the disease, because, it's not only a patient's problem, it is also whoever loves that patient and lives with that patient, and the researchers. And this is a very important relationship, and, communication is very important. I was reading through the chat, the appreciation of, how science is explained and, the clarity. And that's something that, both Dr. Sacher and I, highly regard. Because after all, one needs to keep, knowledge is important. It is important to have as much knowledge as we can. But when we are facing disease, we have to keep the focus on a cure and improving the condition of the patients at the least and cure them at the best. And in so doing, improving also the quality of life of their families that are caring for them. So it's extremely important to keep that communication channel open and active. And this foundation is doing that excellently. So it's really a privilege to to see how active and how open you are to this.
Thank you very much, and congratulations for your awards. And then we will see you soon in person. Thank you very much.
Yeah. Yes, looking forward to that.
Thank you, Giorgio, and congratulations again to all of our awardees this year. As a quick reminder, please save the dates for a few exciting upcoming events. As Anne shared earlier, we have another TANGO2 talk coming up on September 16th, 2003. We have our TANGO2 research symposiums on October 27th. And we have our third family conference, June 23rd through June 25th, 2024. If anyone has any questions, please don't hesitate to reach out to Anne or Dina at the email addresses listed at the bottom of the screen. Thank you everyone again to all of this year's team captains, participants, donors, volunteers, and attendees. We couldn't have achieved and surpassed this year's fundraising goal without all of your efforts to help us shine a light on TANGO2. Thank you everyone, and congratulations through all of your hard work.
Yay, thanks everybody.
Thank you for joining today.
Thanks, everyone.
Congratulations.
Thank you.
Hope to see
Thank you.
everyone soon.
Thanks, everyone. Have a good rest of the weekend.
Thank you.